Hi, my name is Justin Dranschack with Molecular Devices. During today's talk, I will review a workflow for rapid and efficient selection of high-producing mammalian cells secreting non-monoclonal antibody proteins. While there is much to be gained by developing novel therapeutics, the average discovery timeline remains long and laborious. It is estimated that product development timelines from lead target identification to phase 1 IND filing is 10 to 12 months at most companies. Obviously, with the urgency of today's global pandemic and with the necessity of being first to market, the ability to substantially reduce this timeline is increasingly relevant. This is where Molecular Devices' biopharma portfolio can help, by equipping users to substantially collapse a major portion of this timeline that is dedicated to transfecting, selecting, and screening various clones. With Molecular Devices' ClonePix platforms, you can accelerate clone generation and screening timelines by integrating multiple steps into one. The ClonePix is a powerful tool for rapid screening of over 10,000 colonies per run and isolation of top secretory mammalian cells typically used in the production of therapeutic proteins and monoclonal antibodies. The method for screening on the ClonePix starts with the transfection of your cells. These cells are then plated in low density into semi-solid media. Single-cell derived clones then form into adherent or spherical colonies. As they grow, they begin to secrete their specific protein or antibody of interest. These secreted proteins or antibodies then bind to a fluorescent antibody binding detection agent or fluorescent anti-tag, identifying those colonies that are your highest producers. In this example, we begin to explore non-monoclonal antibody methods. Using the ClonePix, we plated HEK 293 cells stably transfected with an untagged monomeric protein of interest. For full-length untagged peptides, the detection of secretion requires a fluorescently conjugated antibody to that protein. In this image, high producer cells are identified using FITC with a cloud of secreted protein around the most productive colonies. Due to the nature of the ClonePix technology, it is important that a polyclonal or several monoclonal antibodies raised against different epitopes to the same protein is used as opposed to a single monoclonal antibody. In the next example, we look at detecting secretors of tag recombinants. For this workflow, with anti-tag fluorescent agent present in the medium, the secreted tag protein and the agent are mutually trapped locally around the colony of cells. The amount of fluorescence is proportional to the amount of protein secreted. The tag must be of sufficient size to be recognized by 2 or more fluorescent molecules so that the precipitation complex can form. Using this principle, we look at detection of secreted monomeric His flag tag protein. His and flag tags were in tandem at the C terminus. 2 tags together were necessary in this case to provide a sufficient tag size for the multi-epitope binding required for formation of the fluorescent complex. Any epitope tag or combination of tags with size greater than 10 or 12 to 14 residues should be sufficient. An example of this would be His or 2X flag. High secretors were then screened and picked into 96-well plates at day 8. Once you have imaged and screened your colonies of interest, the ClonePix software allows you to efficiently and effectively rank your colonies based on several important criteria, such as fluorescence intensity, size, shape, or proximity to other colonies. You can quickly perform a quality control check, and then the instrument will pick your colonies into 10 96-well destination plates for your downstream processes. So in summary, the ClonePix platforms allow you to break away from traditional, inefficient, and time-consuming processes, allowing you to increase efficiency and throughput with a fully automated workflow. Identifying rare clones faster, allowing you to run multiple projects in parallel, and eliminating or recovering unstable clones early in the process, saving you both time and money. At Molecular Devices, we understand customers may have interesting challenges, but we have solutions. Thank you for your time.